Eating food containing Salmonella bacteria can cause illness. Two symptoms of infection by Salmonella are vomiting and diarrhea. What causes these symptoms? This question is asking us about a bacterial infection, so we need to use our knowledge of bacteria to answer this. What do bacteria release that causes you to have this sickness, the vomiting and the diarrhea? And the answer the mark scheme is looking for is toxins. Give 2 ways in which a person with a mild infection of Salmonella can help prevent the spread of bacteria to other people. So this question wants to know, how are we stopping the transmission of the disease? The main answers we're looking for here are to do with personal hygiene. So we can have people washing their hands after going to the bathroom, the infected person after being sick. You can isolate the infected person so they're not interacting with others. And then some alternatives. You avoid the infected person preparing any food, particularly for anyone else. And then you can disinfect everything. So sanitize all the work surfaces and clothing that they've been in contact with. In very serious infections of Salmonella, a doctor can prescribe drugs to kill the bacteria. What type of drug can a doctor prescribe to kill the bacteria? So here we're just using our knowledge. We think what medication is used for bacteria, and that is antibiotics. A person with AIDS may take longer than a healthy person to recover from a Salmonella infection. Explain why. So this question gives us a big clue in the word AIDS, and that's because AIDS stands for Acquired Immunodeficiency Syndrome. And what this means is that there's a problem with their immune system. Their white blood cells aren't properly working to get rid of this illness. So that's what we need to put in our answer. So their immune system is damaged or weaker, and their white blood cells, that's the pathogen-fighting cells in the bloodstream, are less able to combat that Salmonella than they would be in a healthy person. Salmonella bacteria can be transmitted from chickens to humans. Chickens can be vaccinated to prevent the transmission of Salmonella bacteria to humans. Suggest one other way farmers could prevent the transmission of Salmonella from chickens to humans. So we can be isolating the infected chickens, and that's the only thing we need to say for our single mark, but as alternate answers, we can be slaughtering the infected chickens so they can't pass it on. And similar to humans, we can give them antibiotics, again, so they can't pass it on. A restaurant owner employed a scientist to test the effectiveness of 2 kitchen cleaning liquids. The scientist took samples from 2 work surfaces, before the surfaces had been cleaned with cleaning liquids and after the surfaces had been cleaned with cleaning liquids. The samples were then analysed for the number of bacteria they contained. The results are shown in figure 1. So here's figure 1. We have the number of bacteria, and that's the one on the y-axis over here. And then on the x-axis we have the 2 cleaning liquids. So cleaning liquid A is on the left and cleaning liquid B is on the right. So what we're looking at is the difference between the values for before and the values for after. So the before values are the darker grey ones, they're on the left, and the after values are the paler grey ones, and they are on the right. The question's asking which cleaning liquid is more effective, and give a reason for your answer. So looking at the graph again, we want to find out which cleaning liquid, A or B, has a bigger difference in the before and after. So having a look at the values for the sink. So in cleaning liquid A, we're looking at this difference, and in cleaning liquid B, it's this difference over here. So we see that cleaning liquid B has a greater difference. So cleaning liquid B has been more effective in this case. And on the table, the other surface, we can see a similar trend. It's slightly larger on the cleaning liquid B than it is on the cleaning liquid A. So cleaning liquid B is consistently more effective. So here we can write down cleaning liquid B, and for a reason we say there was a greater decrease in the number of bacteria on the surface after we cleaned it. The scientists investigated the effect of cleaning liquid A and cleaning liquid B on the Salmonella bacteria growth in a laboratory. Figure 2 shows the way the investigation was set up. So having a look, we can see we've got the agar, that's our paler grey, and we've got different discs soaked in different substances. So the one down here is cleaning liquid B, up here we've got water, and down there we have cleaning liquid A. The Petri dish was placed in an incubator at 25 degrees C for 48 hours. After 48 hours, the scientists calculated the area around each paper disc when no bacteria were growing. The results are shown in Table 1. So here we have Table 1. On the left-hand side is the discs soaked in different substances, so water A and B, and on the right is the area around the disc when no bacteria were growing. What measurement would the scientists need to take to calculate the area when no bacteria were growing? For this bit, we're actually going to need a formula, because when we look up at our Petri dish, we see the area we're concerned with is a circle. So the formula we need is pi r squared. That's the formula for a circle. So the measurement that we need to take is going to be the radius, that line from the outside of the circle to its centre of the area where there is no bacteria growing. If we just make a note of the radius, now the mark scheme will sometimes allow you to say diameter, so when you've got a circle, radius is at halfway, while diameter is all the way across the circle. So we can have that as an alternate answer, but really you want to say radius. Give one change to the investigation that would allow the scientists to check if the results are repeatable. So here we have this key word, repeatable, and what that means is that the same person can repeat the same experiment more than once and get the same results. So how we test for this is by doing repeats and seeing if we get the same result. So you have to repeat the experiment, do it multiple times and see if our results are similar, and if they are, our experiment is valid. The scientists showed the results to the restaurant owner. Both cleaning liquids cost the same per decimeter cubed, so just one other factor the restaurant owner should consider when choosing a cleaning liquid to use. So again, we have a few things that we can say here, but we only need one of them. So one thing we can consider is the toxicity of the product. We can try and think about whether it's going to be harmful to the restaurant owner or whoever's using it to clean with. Another alternative you could say is how it interacts with other cleaning agents, because you're going to use multiple chemicals. So if it interacts with another cleaner and then makes a toxic byproduct, you're going to have a bit of a problem. And then any other relevant alternative, it could be ease of use or how long it lasts for. The mark scheme gives quite a few to you, so anything else that's relevant to the question. Metabolism is the sum of all the chemical reactions in the cells of the body. One metabolic reaction is the formation of lipids. Give one other metabolic reaction in cells. So for this question, there's a couple of things that you can say, but you only need to say one of them to actually get that mark. So your main one's going to be respiration, but then also for your plant cells, you can get photosynthesis, is quite common. And then just as a couple more alternative answers, you can have the formation or breakdown of most bodily substances. So you can have this breakdown of glycogen in the body, and you can also have the formation or breakdown of proteins. And then the last one is the formation of urea. That's another one that you could say here. Table 2 shows the mean metabolic rate of humans of different ages. So here we have Table 2, and on the left-hand side we've got the age in years. So you can see it's starting from the age of 5 up here, and it is increasing as you go down the table to the age of 45. On the right-hand side, we have the mean metabolic rate, and we have males here and females over here. So this one's multiple choice. It says, what two conclusions can be made from the data in Table 2? Tick 2 boxes. So this one is worth 2 marks. As we go down our list, we want to try and prove the option correct or eliminate it from our list. So let's have a look at the first one. It's saying, as age increases, mean metabolic rate of males and females increases. So it's saying that as we're going down the table, as the age is increasing, it thinks that the mean metabolic rate is actually going up. Well, if we have a look at the males, we can see it's going from 53 to 36, but that's actually decreasing, that's not increasing. And the same with the females, we're going from 53 to 35, so that's also decreasing. So we can say that this one over here is not correct. The next one says males have a higher metabolic rate than females after 5 years of age. So it's saying that after 5 years of age, that row at the top there, the male values are going to be higher than the female values. So if we have a look at the male values, here we have 45 and over here is 42. Well, 45 is larger than 42, so that supports our conclusion. Next one on, 39 and 35, the male one's also larger, so that's good. 37, 35 and 36 and 35. So what we notice, the male values are all larger than the female values, and that does support that statement. So we can say this one's correct and give it a big tick in the box. Okay, so the next one says the mean metabolic rate of females decreases faster than males up to 25 years of age. So here we have that row, the 25 years of age, and the question's saying that the female numbers up until this point are decreasing faster than the male numbers. Well, if we have a look, we're going from 53 to 35 on the females, and on the males we're going from 53 to 39. So the females have decreased by more. And that means that yes, the female numbers have been decreasing faster. So the next one we can tick off and say actually this is correct. So make sure you give that a tick in the box. Now, by process of elimination, we can say that we've actually got our 2 marks. We've found our 2 correct answers. But just to show you why the others aren't correct, so the next one says the mean metabolic rate of males and females decreases more quickly after the age of 35. So this is the age of 35 up here, and it's saying that after this point, the mean metabolic rate of the males and the females, so both of them, are decreasing more quickly. Well, the females is not decreasing. 35 and 35 are the same number. And then the males, similarly, is 37 to 36, so they're right next to each other. So that one we can cross off. That's not going to be correct. And the next one says there is no relationship between age and mean metabolic rate. Well, we can see this already. We've talked about how as the age is increasing, the mean metabolic rate is decreasing. So we immediately know that that one's not correct either. Calculate the percentage decrease in the mean metabolic rate of males between 5 years and 45 years of age. Right, so it gives us this equation over here, and it's saying the percentage decrease equals the decrease in metabolic rate divided by the original metabolic rate multiplied by 100. And it says give your answer to 3 significant figures. So this question is worth 3 marks, so there's 3 steps that we're going to have to go through to actually get to this. The first mark you can get is actually due to you getting the correct numbers into this equation over here. So you've got to figure out what is this decrease in the metabolic rate. And to get this, we have to scroll back up to our table and have a look at the values for 5 years and 45 years on the male side. So here's our table again, and the rows we're concerned with are this one at 5 years and this one at 45 years. And having a look to get the values we want, it's this 53 at the top and the 36 at the bottom, so they're what we're interested in for our equation. So what we've got to do is find this decrease value. So we have to take our numbers from earlier and actually get the difference between them. So 53 was our first value for when they were at 5 years, and 36 was the second at 45 years. So we've got to subtract one from the other to find what we need, and the answer you're going to get is 17. Next, we've got to start plugging in our values to that equation in the green box. So 17 is our decrease divided by the original number. So that is going to be the 53, the value from when they were 5 years old. And then the last thing we've got to do is multiply that value by 100, and that makes it into a percentage for us. So we put that in our calculator, and we're going to get a value of 32.075472, and so on. And it's the calculation that gets you that first mark, and then the second mark is due to actually getting the right answer at the end of it. The final mark you want is for your significant figures. It says here 3 significant figures. So this bit, we've got to take the first 3 values and we've got to make sure that they are correct to 3 significant figures. So we've got 07, so that's going to round up. Our final answer is therefore 32.1. And that gives you your last mark. Regular exercise can increase metabolic rate. 2 people did 5 minutes of gentle exercise from rest. Table 3 shows the effect of the exercise on their heart rate. So here we have table 3. On the left is the time in minutes, and on the right is the heart rate in beats per minute for person R and person S. The question says, describe 2 differences in the response of person R and person S to the exercise. Use information from table 3. So when we're looking at a question like this, where we've got to analyse data, there's really 3 things that you can potentially say. So you can talk about the rate of increase, how quickly the data is changing, and you can also say if there are any plateaus or spaces where the data starts to level off and become the same. And then you can talk about the overall change. Has the data increased or decreased, or has one changed by significantly more than the other? These are what you want to be looking out for. So if we start to have a look at our table, and let's just take rate of increase as our first one to look at. So having a look at the numbers with person R, they're going from 60 to 76 to 85, 91 and to 1. In person S, they're going 78 to 100 to 110 and 119. So we notice in person S, when they're doing that jump from 78 to 100, that's a much bigger jump than the 60 to 76. So for our first potential answer, we can say that person S's heart rate increased at a higher rate or increased faster than that of person R. And make sure you mention both person S and person R to get in your comparison. The next thing we can start to look for is that plateau, some levelling off. And we can see this in person R towards the end of the exercise. So they start to level off at the 99, while person S carries on. So what we can say for that last answer is person R's heart rate starts to level off, and you can include at 99, put some data in there, but it's not necessary for the marks. And then you say with your comparison, person S continues to increase. They're going 119, 129, 132. Right, so now we've made our 2 points and we've got 2 marks, but just to show you the alternative answer, we can talk about the overall change. So in person R is only going from 60 to 99, person S is 78 to 32. So we can add that person R experiences a smaller overall change, or it changes less or increases less than you see with person S. Complete the line graph in figure 3 for person S. You should add a scale to the x-axis and label the x-axis. Okay, so here we've got our figure 3, and we've also got table 3 from earlier just to make it a bit easier to see. So what the question wants us to do is add a scale to and label the x-axis. So that's that one down at the bottom there. And then we've got to plot the points for person S and finish the graph. So let's give a start by trying to figure out the x-axis, and we can do this by using the points from person R that have already been plotted. So if we take the first one, for example, that's at point 60 in the y-axis, and we don't know the x-axis one. But if we find a point in the table that's actually got a 60, we can see the first point here is 60 for person R, and that is at time equals 0. So we know that this bit down here must be 0. And then just to check that the graph is going up in the linear fashion that we want it to, we can check on the next point over here. So we can tell that this must be 70, as it's halfway between the 60 and the 80. And then moving along from the point, we can see that this must be 76, because each of the little boxes is worth 2 beats per minute. And now we can establish that each of those big boxes along the bottom, along the x-axis, represents 1 minute. So let's just quickly fill that in. 1, 2, 3, 4 and 5. That lines up with all of the points. And don't forget to add the label as well, that's time in minutes. Okay, so now we can start to make our points for the person S. So at 0 minutes, the value of beats per minute is 78. So we can make a mark 78, so that's going to be up here. And then at 1 minute it is 100, 2 minutes is 110, and so on. And then now that we've got all of our points, we connect them up together just like they've done with the person R. And then don't forget to label it again, person S. So in this case, your 4 marks are going to be for labelling your lower axis correctly, so the time in minutes bit. You get another mark for doing the scale properly, and then the final 2 are for making sure you plot your person S correctly. After 5 minutes of exercise, the heart rate of person S was 132 beats per minute. When person S rested, his heart rate decreased steadily at a rate of 12 beats every minute. Calculate how much time it would take the heart rate of person S to return to its resting rate. Now, this question is worth 2 marks, and we've got the table down there to help us. So what we want to do here is find out how many beats per minute we have to decrease by to get down to the resting heart rate of person S. So we can find that down in the table here. We can see they're at rest when time equals 0, and so the heart rate is 78 beats per minute. That's what we're trying to get to. So the first one we're going to do is find the difference between that 78 and 132 beats per minute. And now what we need to do is find out how many sets of 12 are in that difference, because each set of 12 takes 1 minute. So by finding how many sets of 12s there are, we can find out how many minutes this transition here is going to take. So 132 minus 78, divide that by 12, and that's your first mark for getting it into the formula over here. And the second mark is just for getting that correct answer, so stick that in your calculator. You're going to get 4.5. A student made the following hypothesis about the heart rate of smokers and non-smokers during exercise. During exercise, the heart rate of smokers increases more than the heart rate of non-smokers. Design an investigation that would allow you to test this hypothesis. So this question is worth 6 marks, and when you've got a 6 marker, it's not only about getting all the points down, it's about actually making sure they're sequenced in a logical order. Now, this question concerns 2 different groups of people. We have the smokers and we have the non-smokers. So the first thing that the person's got to do for their experiment is get the 2 groups of people and separate them into smokers and non-smokers. And they've also got to make sure that their groups are of a sensible size, because if you haven't got enough people in your investigation, the results aren't valid. So each group has to be a minimum of 5 people strong. The next thing the student has to do is get these 2 groups of people to carry out the same exercise for the same amount of time, because this is how you're going to get their heart rate to go up. But you must list those controlled variables to actually make sure that it's a fair test, otherwise your results are not going to be valid. So the control variables in this instance are going to be the fact that they're doing the same exercise and the fact that they're doing it for the same amount of time. And they can be listing a couple more controlled variables as well. The key ones the mark scheme is going to have you going for are making sure that the groups are all of the same age and that they're all of the same approximate health, so the same fitness, and that they're the same gender. Now we're going to go on to the dependent variables. So this is the factor in the experiment that we're going to be measuring. And because this experiment is about the heart rate, it's that we've got to be measuring. We have to measure the heart rate of each person, both before and after they carry out this exercise. And then the last thing that they've got to do is a little bit of analysis of this data. So they've got their values for before and after the heart rate. Now they need to calculate the difference between the two so that we can see how much each group's heart rate has actually increased. And they've got to do that for each person so that they have enough data to actually carry out a valid comparison. So we've got a minimum of 6 points here, probably more. But just to check, you want to try and organise your thoughts so that you have 6 points that you're making to get those 6 marks. So we've got our 2 groups of people, that's the mark. Each group have at least 5 people, so we're stating the numbers, so you can get another mark there. Both groups doing the same exercise for the same amount of time, that's at least 1 more mark. And then again, we're listing our controlled variables, that's another down here. Measuring the heart rate before and after, that's the 5th mark. And then a sixth mark would be calculating the change in each person's heart rate. But another important mark is comparing these results. That's an important one to get. So we've got 7 points. You only need 6, but make sure you are getting these bottom two here and at least a controlled variable and your dependent variable. They're the most important things that you can get in the investigation. The circulatory system is composed of the blood, blood vessels and the heart. Urea is transported in the blood plasma. Name 2 other substances transported in the blood plasma. Your answer will be worth 2 marks. So there are multiple answers that you could give for this question, we only need 2. And the first 2 that you're probably going to want to go for are carbon dioxide and water. However, just to give you a couple of alternatives, you can also say glucose, amino acids, and you've got your lactic acid as well. Some athletes train at a high altitude. Training at a high altitude increases the number of red blood cells per centimetre cubed of blood. Explain why having more red blood cells per centimetre cubed of blood is an advantage to an athlete. Now this question is worth 3 marks, meaning there's 3 points you're going to have to make in your explanation. So the first point that you can make is that you've got more red blood cells per centimetre cubed in your blood, and you've got to think about what that means for the athlete. So what does the red blood cell actually do? Well, it contains hemoglobin, and hemoglobin is the chemical that bonds to your oxygen. It enables the blood to carry that oxygen. Which leads us pretty nicely onto our second mark and the second point that we're going to make, and that's that there's more oxygen being transported in the blood now because of that extra hemoglobin. And then the last mark, the last point we're going to make is about what we're actually doing with that extra oxygen that the athlete has acquired. And oxygen is used for respiration, so more of it allows for more aerobic respiration of the muscle cells, and this enables them to do more activity because the muscle cells have more energy from that respiration. Note this keyword aerobic respiration here. So this is the term that we use for respiration that has got the presence of oxygen. Which 2 blood vessels carry deoxygenated blood? Tick 2 boxes. So we've got 2 marks over here. There's 2 that you've got to tick. Make sure you get both. Just as a reminder, we're talking about deoxygenated blood. So this is blood without oxygen. And this tells you that the blood is on its way to the lungs rather than from the lungs. So that can help us out a little bit here. So if we have a look at the first one that's on our list, that is the aorta. And so we want to be thinking about what this one actually does, which is deliver blood from the heart to the rest of the body. And it's delivering oxygen, which means that the blood coming through the aorta must be oxygenated, so we cross that one off. The next one is the coronary artery, and thinking about this word coronary, that means that it's to do with the heart. And what it's doing is delivering oxygen to the heart, so we can cross that one off and have a look at the next one. So this one is the pulmonary artery, and the fact that it's an artery tells us that it's actually delivering blood away from the heart. However, if the aorta is taking the oxygenated blood away from the heart to deliver to the body, this pulmonary artery must be on the second loop, where the deoxygenated blood has already come back to the heart, and now it needs to go to the lungs. So the pulmonary artery delivers deoxygenated blood to the lungs. So yes, we can tick this one. It is deoxygenated. Then we have the pulmonary vein. So veins are those that are delivering blood to the heart. And this one is taking your oxygenated blood into the heart. This one's the one that comes from the lungs. So we can cross that one off because it's already gone to the lungs and got some oxygen. So that's going to be oxygenated. Next one, the vena cava, comes in on the right side of the heart and it's delivering the blood that's been around the body and given up all of its oxygen. So it's taking that one back to the heart. And it will be deoxygenated because it's the blood that wants to go to the lungs. Figure 4 shows 3 types of blood vessel. Which type of blood vessel carries blood into the right atrium? Tick 1 box. Right, so the key thing in this question is it's asking us which blood vessel carries blood into the right atrium. And we know that the blood vessel carrying blood into the heart is always going to be a vein. So what the question is actually asking us is which of these 3 blood vessels is a vein. and we've got to think about our characteristics of blood vessels to actually figure this out. So if we have a look at blood vessel A, we can see that we've got a very narrow lumen, and we have a very thick muscular or elastic wall. And what this tells us is that blood vessel A is carrying blood at very high pressure, and it has to be able to withstand this. That's why there's so much muscle and so many elastic fibres. So blood vessel A is actually what's taking the blood away from the heart. Blood vessel A is an artery. Looking at blood vessel B, we can see that there's a very wide lumen. And then over here we have a much thinner wall or thinner layer of muscle and elastic fibre. And blood vessel B is therefore at a much lower pressure. So this is the one that's actually taking the blood back into the heart. So this is the one we're looking for. Blood vessel B is the vein. And then just for your information, blood vessel C, we can already tell, is really small. And we can also see over here, it's only 1 cell thick. And this is because it's capillary, and the 1 cell thickness allows for better diffusion. Anyway, the answer that we're looking for is B. That's the vein that's carrying blood back into the heart. So we tick that one. Compare the structure of an artery with the structure of a vein. And this question is worth 3 marks, so there are 3 points that you're going to have to make. Now, it's really important here that you notice this word compare, because this tells you what kind of language you need to be using. So there are 3 things that you can really be talking about. You can talk about the wall, and then you can talk about its elasticity and thickness. You can talk about the size of the lumen, and you can talk about if there are valves or not. So if we take the first one and talk about the walls, we know with our arteries, they're going to be carrying blood at much higher pressure. They're taking blood away from the heart. And because they're at a higher pressure, we know that it's going to be a lot more muscular. So we say that arteries have a thicker layer of muscle. And make sure you get this thick bit, because that's your comparative language. And if you want to, you can finish it off with than veins, again, just to add to the clarity for the examiner. Then if you look at the size of the lumen, we can say that the lumen is narrower, and again, this is because we've got that really thick wall. And that's 2 marks that we've got here. The third mark you can get by talking about the valves, because when we're talking about veins, we know that they're under a lower pressure, and there's more chance of the blood flowing backwards, so this is called backflow, and the veins have valves to prevent this, whereas the arteries do not. What you could also add, just as an alternative mark, is that the arteries have thicker elastic tissue. That's all you really need to point out. So we've made 3 points, 1, 2, 3, and then that is an extra one if you want it. You don't need all 4 to get the 3 marks. Heart rate is controlled by a group of cells. This group of cells act as a pacemaker. Figure 5 shows a section through the heart. Draw an X on figure 5 to show the position of the pacemaker. So let's just clarify this term pacemaker. What it is, particularly in an artificial context, is a device that helps the heart to beat rhythmically, so it helps you deal with arrhythmia, and it will correct any abnormal rhythms. And so in your heart you have a group of cells that set that rhythm and make sure your heart's beating nice and steadily. So here we've got figure 5, it's a cross section of the heart, and you've just got to draw an X where the pacemaker goes. And according to the mark scheme, your little X marking the pacemaker can go in anywhere in that yellow area there. So for example, we might just put ours here. A patient may be fitted with an artificial pacemaker. What condition may be treated using an artificial pacemaker? So for this question, you only need to say one thing, and you don't need to name a specific condition. You can just say an irregular heartbeat. And you can say arrhythmia, which means the same thing. A student carried out an investigation using chicken eggs. This is the method used. Place 5 eggs in acid for 24 hours to dissolve the eggshell. Measure and record the mass of each egg. Place each egg in a separate beaker containing 200 centimeters cubed of distilled water. After 20 minutes, remove the eggs from the beakers and dry them gently with a paper towel. Measure and record the mass of each egg. and table 4 shows the results. So here we've got our table 4. On the first column on the left we have the eggs, so they've been numbered 1 through 5 to identify them. Then in the next column along we've got the mass of the egg without the shell, and then on the last one it's the mass of the egg after 20 minutes in grams. So you can think of it as egg number, mass before and mass after. Another student suggested that the result for egg 4 was anomalous. Do you agree with the student and give a reason for your answer? So we have a copy of the table up in the top left, and what the question's asking us to do is identify if this row here is anomalous. The way that we will go about this is look at the difference between the values in this column over here and this column over here. So we want to calculate the differences. So if we just go through each of these rows and calculate the difference between the latter one and the former one. So here I've done 77 minus 73.5, and that's given me a difference of 3.5. The next one, 73.9 minus 70.3, gives us 3.6. And we're going to repeat this calculation going down the table. So 3.3, 1.5, and 3.3. Right, so having a look at these numbers, we can actually see that egg 4 is 1.5, while the rest seem to be ranging from 3.3 to 3.6. So actually it does look like egg 4 is anomalous. So for our answer we can write yes, egg 4 is anomalous, because the mass change is a lot lower than in the others. Calculate the percentage change in mass of egg 3. So this question is actually a little bit mathematical. There's a formula that you're going to need to use to be able to calculate this percentage change. And that formula is your final value minus your initial value, and then dividing again by that initial value, and then multiplying by 100 to make it a percentage. So here we've got our table 4 again. We're concerned with this third row down here for egg 3, and the values we want are 72.4 and 75.7. And this one here is the final value, and there is the initial. So let's just plug those into our formula. We're doing 75.7 minus 72.4, and then we divide that by 72.4, so that's our initial value. And then we will multiply by 100 to make it a percentage. So we calculate that and get an answer of 4.6%. So here you get 1 mark for doing this calculation and another mark for getting the correct answer. Explain why the masses of the eggs increased. Now, what we've got to remember here is that the eggs have just spent 20 minutes sitting in water, and so what's actually happened is the water has moved from that surrounding solution around the egg into the egg itself. That's why the mass has increased. And this movement of water across the partially permeable membrane we call osmosis. That's the key term for this question. And the first mark available for you to get is actually stating this term. So we say the mass increased because water entered the egg by osmosis. Then for the next 2 marks, as this is a 3-mark question, we have to define this term osmosis. And the way that we define this is by saying that the water has moved from the dilute solution in the beaker to the more concentrated solution in the egg. So that gets you your 2nd mark. And then for your 3rd mark, the last point you have to make is mentioning the partially permeable or selectively permeable membrane. And if you just have another look at this term, partially permeable means that only certain substances can actually get through this membrane. So in this instance, we're talking about the water crossing into the egg. Explain how the student could modify the investigation to determine the concentration of the solution inside each egg. And this question is worth 3 marks, so there's 3 points that you're going to have to say. So with this investigation, we're looking at different concentrations. So the first thing that we actually want to point out is that we're going to use 5 different concentrations of sugar solution or salt solution. And we say 5 because this is really the minimum number that you need to actually plot a graph and get a reliable trend. And what we're going to be doing with these different solutions is we're going to put the egg in there like what we did before with the water in the last investigation. Only this time we've got different concentrations. So we're going to measure the masses of the egg before and we're going to measure the masses of the egg afterwards. and we're going to calculate the percentage change. So we can plot that on a graph and see if we get a trend. And what we're looking for is the point where there is 0% change, because when there's 0% change, it means that the concentration is equal on either side of the membrane, and that's where there's no osmosis occurring. So what we need to put for our answer is that we're determining the concentration where the curve crosses the 0% change. Chicken eggshells contain calcium. Calcium ions are moved from the shell into the cytoplasm of the egg. Table 5 shows information about the concentration of calcium ions. So just below we can see we've got table 5. In the left hand column it's telling you where it is, so if it's in the eggshell or the cytoplasm, and on the right hand side it's telling you the concentration of the calcium ions. The question is asking you to explain how calcium ions are moved from the shell into the cytoplasm of the egg. Now, if we look at the table, we see the concentration of ions in the eggshell is 0.6, and in the egg cytoplasm it is 2.1. The question's told us it's going from the shell to the cytoplasm, it's going from here to here. And looking at these numbers, we realise that it's going from an area of lower concentration in the 0.6 into an area that is actually more concentrated at 2.1. And this combination, the area of lower concentration to an area of higher concentration, is known as active transport. And to get the marks in this question, we have to state that we're talking about active transport, and then we have to explain it. So the first mark that we can get is actually for what we've just gone over. So the idea that the ions are moving from an area of lower concentration into an area of higher concentration. We say that this is done by active transport. And then that third mark is just for knowing that active transport requires energy, which makes sense because we're going against the concentration gradient. Plants can be infected by fungi, viruses, and insects. Aphids are small insects that carry pathogens. Figure 6 shows an aphid feeding from a plant stem. So here's figure 6, and the insect is the aphid, and we can see its sharp mouthpiece is just piercing the stem there. So the question says, an aphid feeds by inserting its sharp mouthpiece into the stem of a plant. Give the reason why the mouthpiece of an aphid contains a high concentration of dissolved sugars after feeding. So the key thing about this is that it is feeding, and it's inserting that sharp mouthpiece into the stem of the plant. And what it's actually trying to get to is these phloem tubes that are running inside the stem of the plant, because they're what's transporting all of the sugary substances that the plant is actually using for itself. And so the result is going to be that mouthpiece having a high concentration of dissolved sugars. So we say for our answer that the aphid has been feeding from the phloem of the plant. And ensure that you write down this key term phloem. Plants infected with aphids may show symptoms of magnesium deficiency. Magnesium deficiency symptoms include yellow leaves and stunted growth. Explain how a deficiency of magnesium could cause these symptoms. And this question is worth 5 marks, so there's 5 points you've got to make. So the questions actually helped us out by giving us these 2 points over here, but we also have to use some of our knowledge of the nutrients. So magnesium is what helps the plant to make chlorophyll. Chlorophyll being the pigment that gives the plant its green colour and helps it with photosynthesis. So when we've got less of it, we see that symptom of the yellowing leaves. And that's what we say for our first point, that the yellow leaves is caused by a lack of chlorophyll. Now, the way that chlorophyll helps with photosynthesis is it absorbs the sunlight from the external environment so that the plant can then turn into glucose. So the second point is that less light is therefore absorbed by that chlorophyll. And you get another mark for saying this results in a lower rate of photosynthesis or less photosynthesis. Why is this less photosynthesis a problem for the plant? Well, it means that the plant is making less glucose. Plants need glucose for a number of things. It has a lot of uses for them. One is that less glucose will then be converted into protein, and that means that the plant can't grow properly. It can also alternatively mean that less glucose will be converted into cellulose, and this is a main component of the cell wall, and it's really important for the plant and its rigidity. The last thing is that glucose is also used to provide the plant with energy, and the plant needs energy to carry out protein synthesis. So any of these can be your last mark, but the point that you actually want to be getting up is any one of them will result in that symptom of stunted growth. They'll stop the plant from growing properly. A farmer thinks a potato crop is infected with potato virus Y, PVY. The farmer obtains a monoclonal antibody test kit for PVY. To make the monoclonal antibodies, a scientist first isolates the PVY protein from the virus. Describe how the scientist would use the protein to produce the PVY monoclonal antibody. And this question is worth 4 marks. So we're going to try and break the process down into 4 steps and 4 points. The first of them being injecting the protein into a mouse. Then the next thing that needs to happen is combining the lymphocytes, and a lymphocyte is just a type of white blood cell, a type of pathogen fighting cell, and they're combining that with the cancer cells. And they're doing that so that they can start to make these hybridoma cells. So a hybridoma, you can kind of identify it by looking at the first section of the word hybrid, and it just means it is a fusion of that white blood cell with the cancer cells from a tumour. So the scientists want to try and identify a hybridoma that will be able to produce monoclonal antibodies that are specific to that PVY virus. And then once they've found it, they're going to clone it so they have many cells that are actually making this monoclonal antibody. Cystic fibrosis is a genetic disorder caused by a change in a gene. What molecule are genes made of? So it's essentially asking what's our genetic material, and that's DNA. And if you want to write it out in the long way, you can say deoxyribonucleic acid. Cystic fibrosis affects the cell membranes of cells in the lungs and in the digestive system. What is the function of the cell membrane? And the answer that the mark scheme is going to be looking for here is that it controls the movement of substances in and out of the cell. In a person with cystic fibrosis, cells lining the lungs and digestive system create too much mucus. The mucus can block the duct leading from the pancreas to the small intestine, and it can block the tubes leading to the alveoli in the lungs. Explain why children with cystic fibrosis grow more slowly than children without cystic fibrosis, and this question is worth 6 marks. Which means that in addition to giving your relevant points, you've got to make sure you're linking them together in a logical structure and explaining in enough detail that the examiner knows you know what you're talking about. So this question wants us to talk about the pancreas, which releases your digestive enzymes into the small intestine. And then we're also talking about the alveoli, which allows a diffusion of oxygen from the lungs into your bloodstream. So if we first talk about the pancreas, we've got our pancreatic duct between the pancreas and the small intestine, but that's been blocked by mucus. And the problem posed here is that fewer digestive enzymes can get through from the pancreas into the small intestine and start breaking down the food. So for our first mark, we have to state this. We have to say that the enzymes can't break down the food. And then our second mark is the result of this. So if we can't be breaking down the food properly, the body isn't going to be able to absorb the nutrients that it needs. And then the next mark that you get is specifically for mentioning the amino acids, because we need those to be in the bloodstream so that we can then start to build our proteins. And so that's going to get you your 3rd and 4th mark. You say fewer amino acids are entering the bloodstream, therefore we're making less protein, which is needed for growth. Now, because this is a 6-mark question and it's given us these 2 bullet points, the pancreas and the alveoli, we need to make sure that we're mentioning both, otherwise we won't get all of the marks. So what we've just said, the first 4 bullets, are all regarding the pancreas. So now we need to start to speak about the alveoli. And as we mentioned earlier, the alveoli are responsible for allowing that diffusion of the oxygen from the lungs into the bloodstream. So if the tubes between the alveoli and the lungs are blocked, then we've got less oxygen entering the blood. And this is a problem because the cells need the oxygen to be there so that they can carry out respiration, and that's your next mark. And that leads you nicely onto your last point, which is why you need the respiration. And that's because respiration provides you with energy, which the body needs for growth. And now you've got your overall reason of why the child with cystic fibrosis is growing a bit slower than normal. Table 6 shows information about people in the UK in 2015. So here we've got our table 6. In the left-hand column, we've got people with cystic fibrosis and the whole population. So there are 2 rows. And then you've got on the right-hand side, median age in years. And the question is saying, describe how the median age of a group of people can be determined. And you get 2 marks for this one. So the median value is the middle value. And to identify the middle value in a range of data like this, we have to put the ages in chronological order. And doing that will get you your first mark, just making that statement. The second mark is then for stating that you have to find the middle value, because that's going to give you your median. Suggest one reason why the median age for people with cystic fibrosis is lower than the median age for the whole population. So there are a couple of answers you can potentially give for this. One is that cystic fibrosis is most common among the younger population, so the entire range of data has shifted towards the younger people, which lowers the median. Or you can go for the alternate answer, which is to say that more people don't make it to the older age when they have cystic fibrosis. They don't live all the way into adulthood. People with a lung function below 30% may need a lung transplant. Table 7 gives information about people with cystic fibrosis in 2015. So here we've got Table 7. The left-hand column is talking about the lung function, and then on the right-hand column we have the percentage of people with cystic fibrosis. In 2015, the total number of people with cystic fibrosis in the UK was 10,800. Calculate how many people with cystic fibrosis in the UK in 2015 would not need a lung transplant. And over on the left-hand side, we've actually got a copy of Table 7 to help us out. So when we're tackling this, we need to remember that it's the people with below 30% lung function that need the transplant. So it's that bottom row there that we're talking about. And the table's telling us that it's 2% of the people with cystic fibrosis that are needing the lung transplant. But remember, the question's asking how many do not need the lung transplant. So what we've got to do is get our 100%, so that will be all people with cystic fibrosis, take away the 2% that do need the lung transplant, and that's going to give you 98%. And then we make that into a decimal so that we can carry out a calculation with it. So that's going to be 0.98. And we use that decimal to find 98% of the 10,800 we had at the beginning, because that's 100%. So the way that we do this is by multiplying the two together. So we do 0.98 multiplied by 10,800. And that's going to give you an answer of 10,584. Lung transplants from donors have risks. One risk is organ rejection. Scientists are researching how to solve the problem of organ rejection and hope to use stem cells to create healthy lungs. The healthy lungs can then be transplanted into cystic fibrosis patients without the risk of organ rejection. Describe how scientists may use stem cells to create healthy lungs that are not rejected by the cystic fibrosis patient. And this question is worth 4 marks. So in order for the patient to not reject the new set of lungs that they're being given, the set of lungs has to be an organ match for them. And the way that we can make it an organ match is by using the stem cells of the patient himself. And these can come from the patient's bone marrow or even from their skin cells. And now that we've acquired these cells, we need to remove the faulty gene, the cystic fibrosis gene, because this patient is a sufferer of cystic fibrosis and we don't want to give them a pair of lungs that also have the disease. And then the scientists are going to create an embryo using these corrected stem cells. And that paves the way for the last step, which is removing these stem cells from the embryo. So these are called embryonic stem cells. And they're going to encourage the differentiation into lung cells. So differentiation is just when the cells specialise into the kind of cell that they're going to be in the body in the future. And that's your fourth point right there. You don't need to say anything else. So each of the statements will get you one of those marks. Some people disagree with the use of stem cells because of the risk of cancer. Give one other reason why some people disagree with the use of stem cells to create new organs for transplants. So what you want to be talking about here is the idea of ethical or religious concerns, because an embryo can be considered a human life or a potential human life. Table 8 shows information about some food components in cow's milk. So here we've got our table 8. The far left-hand column has got the different components. So it lists energy, fat, salt, calcium and vitamin B12. Then in the next column along, so the middle column, it's got the value per 500 centimetres cubed. And then the last on the right hand side is the recommended daily allowance for a typical adult. How much more milk would a typical adult have to drink to get their recommended daily allowance for calcium compared with the amount of milk needed to get their recommended daily allowance for vitamin B12? And we got a copy of the table 8 in the left to help us out, and this question is worth 3 marks. So if we talk about the calcium first, we know that we've got 500 centimetres cubed, and in that we have 605 milligrams of calcium. So what we want to do is find out the volume of just 1 milligram of calcium. And the way we do that is by getting the total volume, so the 500 from that 500 centimetres cubed. We divide that by the number of milligrams, 605, so that gives us the volume of 1 milligram. And then we multiply that by 1,000 because we know we need 1,000 milligrams. So that's going to give us that total volume that we're looking for. And the value is 826.446281 centimetres cubed. That's how much milk we need to get our recommended daily allowance for calcium. And we repeat this to figure out how much milk we need to get our RDA for vitamin B12. So 500, that's the volume of milk we've got. We divide that by the value of vitamin B12 inside that, so that's the 4.5, you get that from the bottom row in the table, multiplied by the recommended daily allowance, that's 2.4, and you get 266.67 centimetres cubed. So that's how much milk we need to get our recommended daily allowance of vitamin B12. And remember, the question's asking us how much more milk we need to get our recommended daily allowance of calcium than we do to get our allowance of vitamin B12. So we're going to get our value for calcium, 826.446 and so on. And then from that we take away 266.67 and that gives us 559.77 and so on. And we round that to a sensible number of significant figures. So we've put 560, and there you've got your 3 marks. So one for calculating a value for calcium, another for the vitamin B12, and the final for getting that last answer at the end. Describe how a student could test cow's milk to show whether it contains protein and different types of carbohydrate. And this question is worth 6 marks. So this question wants you to talk about the different types of food tests that you can do. And if we start with the protein, you need to be mentioning the burette reagent, or you can mention copper sulfate or sodium hydroxide. However, the mark scheme will immediately want you to go for the burette reagent as a rule. And this is how you test for protein, and that will get you your first mark. And because this is a 6-mark question, you've got to make sure that you're explaining your statements as well in a logical order. That's what gets you those marks. So you then say that you add the burette reagent to the milk, that's another mark, and you describe the colour change that will happen. So the solution will turn from blue to a lilac colour if positive. And positive just means that the thing you're testing for is present. Now we can start to move on to talking about carbohydrates. So if we start to mention the test for starch, that is done with an iodine solution. So you make that statement, iodine solution test for starch, so that's another mark there. And like what we did with the burette reagent, we explain what you do with it and what the colour change will be. So in this instance, we're adding the iodine solution to milk, there's your next mark, and then you say this colour change. So the solution will turn from an orangey colour into a blue-black colour. So always be describing the result for the positive test. Now you should be aware that the question has asked for different types of carbohydrates, so we don't just want to talk about the starch, we can also talk about glucose as another type of carbohydrate, and what you're using there is the Benedict's reagent. So your next mark can be Benedict's reagent tests for sugars, and then again, same as before, we describe what we're going to do with it. So in this instance, we add it to the milk, and then we're actually going to heat it up a little bit, so around 60 degrees C is what you could say. And then in terms of the colour change, there is a little bit of a difference because it could have many different outcomes. So you can state the starting colour of the Benedict's reagent, which is blue, and then any of the colours that are listed down here, the mark scheme will allow you to say. So this could be red, brown, orange, and it could be yellow or green as well. So there are a lot of potential ones over there. Now you'll notice that we've got more than 6 points written down here, but you should be aware that just making 6 statements won't necessarily get you all of those marks. You've got to make sure that you're explaining everything to the examiner if you want to get full marks in this type of question. And you want to have a logical structure like how we've worked through each of the components here. A scientist investigated the effect of bile on the breakdown of fat in a sample of milk. The scientist used an indicator that is colourless in solutions with a pH lower than 10 and pink in solutions with a pH above 10. This is the method used. Add 1 drop of bile to a test tube and 1 drop of water to a second test tube. Add the following to each test tube: 5 centimetres cubed of milk, 7 centimetres cubed of sodium carbonate solution, 5 drops of indicator, 1 centimetre cubed of lipase, and then time how long it takes for the indicator in solutions to become colourless. The results are shown in Table 9. So here's Table 9, and the left-hand column is just telling you what's in each row. So there's the solution with bile in the top and the solution without bile at the bottom. And then in the right-hand column, it shows the time taken for the indicator to become colourless, measured in seconds. And the question's asking you to explain why the indicator in both tubes became colourless, and it's worth 3 marks. So to go about answering these questions, we can start to talk about the effect of the lipase, because lipase is a digestive enzyme, and what it does is break down the fats into fatty acids and glycerol. So that will get you your first mark, just making that statement there. The second mark you get for explaining that a little bit further. So what is the result of this? And the answer is that the fatty acids lower the pH, so that's the second mark there. Then the last one, you explain why that causes the indicator to change colours, and that is because the fatty acids cause the pH to drop below 10, and that will cause the indicator to become colourless. That will trigger that change in both of the tubes regardless. And the explanation behind this is just because of those fatty acids. The acids have the pH of less than 7, and they have the effect of lowering their overall pH. Give the reason why the measurement of the time taken for the indicator to become colourless might be inaccurate. So the idea that the mark scheme wants you to get at here is that the observation of this colour change that is happening with the indicator is subjective, so it's opinion based. And what is regarded as a colour change by one person may not be by another. Explain the difference in the results for the last 2 test tubes in Table 9. This question is worth 3 marks. So we have a copy of Table 9 in the bottom right here, and we can see that the solution with bile takes 65 seconds and without takes 143 seconds. So what the question's really asking is, why did the solution without bile take longer than the one with bile? And the reason for this is that bile will emulsify fat. And if we take a look at this term here, emulsifies, what that means is that it's breaking those larger droplets into smaller ones that are individual. So it just gets the larger ones and makes them into smaller ones. And the result of this is a change in that surface area to volume ratio. So the surface area gets larger, because those fat droplets are smaller, and that then makes it much easier for the enzyme lipase to break down the fats into fatty acids because there's a lot more surface area accessible to it, so it can carry out the reaction faster. And it's got the fatty acids appearing before the solution that doesn't have bile, and so the indicator is going to change colour before the other one. And that's as far as you have to explain, so each statement is worth a mark to get you those three.